Hello everyone. Today we will discuss about a new topic that is nucleic acid. Nucleic acid क्या है? Nucleic acids are the are basically of देखा जाए तो nucleic acids are of two types. Okay, nucleic acids are basically of two types. Deoxyribonucleic acid that is DNA and ribonucleic acid. RNA. These nucleic acids are basically act as a genetic material. Act as a genetic material. Okay. These nucleic acids are of these nucleic acids are polymer of न्यूक्लियोटाइड्स. ये सब न्यूक्लियोटाइड्स का पॉलीमर्स है. ओके. सो फर्स्ट न्यूक्लिक एसिड was discovered by Friedrich Miescher from the nucleic acid of past cells, that is leukocytes. Okay. Now the nucleotides, these are the I told you that nucleic acids are polymer of nucleotides. Then what is nucleotides? Nucleotides are nucleosides plus phosphates, and these nucleosides are divided into two parts: बेस, जिसको हम बोलते हैं नाइट्रोजिनस बेस एंड सुगर. Okay. These nitrogenous bases are of two types. These nitrogenous bases are of two types. One is purine, another one is pyrimidines. Purine, that is adenine and guanine, but in case of pyridine there is some difference there, that is for DNA it is cytosine and thiamine. For RNA it is cytosine and uracil. Okay, These are the important point. You have to remember that the nucleic acids are the polymer of nucleotides. Okay, then basically nucleic acid of two types, that is deoxyribonucleic acid and ribonucleic acid, and both are act as a genetic material. So first nucleic acid was identified by Frederick Messner from the nucleic acid of POS cells. The nucleic acid are the polymer of nucleotides. Then nucleotides are composed of nucleosides and phosphates. Nucleosides again divided into two parts, that is nitrogenous bases and sugars. Nitrogenous bases are again of two types, that is purines and pyrimidines. For purines, adenine guanine is both common for DNA RNA, but the pyrimidine is different in case of DNA and RNA. For DNA, pyrimidine is cytosine and thiamine, and for in case of RNA, it is cytosine and uracil. Okay. Now we have discussed about the bases. Okay. Now let us discuss about sugars. So the natural occurring sugars in DNA, RNA are measure of two types. That is, these are basically pentose type of sugar. Okay, that is ribose and deoxyribose. Okay, ribose is basically found in RNA. Ribose is found in RNA and deoxyribose is found in DNA. So basic difference between ribose and deoxyribose is one hydroxyl group. Okay, at second position there is a presence of OH group in case of RNA, but same OH group is absent. Second position OH group is absent in case of DNA. Okay, so due to this, and this OH group is basically replaced by hydrogen group in case of DNA. Okay, so everyone is clear up to this. Then important point is this pentose sugar binds to nitrogenous bases. Pentose sugar binds to nitrogenous bases through a linkage, that is beta N glycosidic linkage. By help of beta N glycosidic linkage. Okay, so everyone is clear up to this. Now let us discuss about structure of DNA. So DNA is a having double helix structure. Double helix means like this, or double strand structure. Okay, one one strand is from 5 prime to 3 prime direction, another one is 5 prime to 3 prime direction. Okay, the both strands of DNA are anti-parallel to each other. Okay, these DNA having three major forms, that is bDNA, B form, zDNA and aDNA. Okay, here is a exception. bDNA is right-handed orientation, but there is an exception. B and A DNA have right-handed structure, but this Z DNA having left-hand orientation. Okay, okay. The double helix DNA structure is basically proposed by Watson and Crick. You should remember. The double helix structure of DNA is proposed by Watson and Crick. And this B form of DNA is predominant under physiological condition of low ionic strength. It is predominantly present in physiological condition at low ionic strength. Okay, this double helix DNA having diameter around 2 nanometer. Okay, and each turn each turn of the helix is around 3.4 nanometer, which comprise of 10 base pairs. Okay, then there is a formation of bond between AT and GC. Adenine forms bond with thymine, guanine forms bond with cytosine. That, but there should be important points to note that adenine having two bonds, whereas guanine and cytosine having three double bonds. Okay, three hydrogen bonds. It is having hydrogen bond, AT having two hydrogen bond. Okay, then the double helix model DNA follows Chargaff's rule. The double helix DNA, only the double helix DNA structure follows the Chargaff's rule. So first point in Chargaff's rule is concentration of adenine is equal to concentration of thiamine and concentration of cytosine is equal to concentration of guanine. Okay. What Chargaff's rule tells that the concentration of adenine is equal to concentration of thiamine. The concentration of cytosine is equal to the concentration of guanine. Okay. Second point in Chargaff's rule is base composition of DNA from different species varies with. Okay. The concentration of adenine is equal to concentration of cytosine. It is the first priority. It is the first priority of the rule. Okay. Third point is the second priority of the rule, that is in a single strand of double stranded DNA, in a single strand of double stranded DNA, percentage of A is nearly equal to percentage of T, then percentage of C is nearly equal to percentage of G. This is the second priority of this rule. Okay, this is the major important point associated with the Chargaff's rule. Initially we discussed about B form of DNA. Let us discuss about Z form of DNA. Z form is left-handed double helix model with 12 base pair. In B form there was a 10 base pair, but here there is a 12 base pair. Okay. Then important concept is denaturation of DNA. So DNA denaturation basically depend upon few important factors such as change in pH, then increase in temperature. These two factors, that is change in pH and increase in temperature, causes separation of that leads to separation of double strand. Okay, due to this separation, the separation of double strand of DNA, that called as denaturation. Okay, due to this the helical structure of the DNA breaks. The temperature at which one important concept is Tm, that is melting point temperature. What is temperature at which half of helical structure temperature at which half of helical structure of DNA is lost. That temperature is known as Tm or melting temperature. Temperature at which half of the helical structure of DNA is lost. That is known as melting temperature. Okay. So the melting temperature basically depend upon the GC content of the DNA. जिस डीएनए में जीसी का कंटेंट मतलब ये डायरेक्टली प्रोपोर्शनल है. क्यों? इसका रीजन भी मैं बताता हूँ. क्योंकि ना जीसी हैविंग 3 हाइड्रोजन बॉन्ड whereas AT having 2 hydrogen bond. There is more temperature required to break the GC bond. That's why the content of GC is responsible. The content of GC is one of the important factor or most important factor for the TM. So the TM is basically depend upon the GC content of the DNA. Okay, let us discuss about RNA. RNA is a single stranded polymer of ribonucleic acid joined by phosphodiesterase bond. Or specifically 3-5 phosphodiesterase bond. Okay. The second position hydroxyl group is highly susceptible for alkaline hydrolysis. Highly susceptible for alkaline hydrolysis. And one important point is the ribose moiety present in RNA show color reaction with original. So this point is very much important. RNA the ribose moiety present in the RNA produce color reaction with original. The 2' hydroxyl group basically highly susceptible to alkaline hydrolysis. This is at RNA at the single stranded polymer of ribonucleic acid which is joined together by phosphodiesterase bond. Okay. So there are 3 major types of RNA. 3 types of RNA. What are they? These are mRNA, second one is tRNA, third one is rRNA. Let us discuss one by one. mRNA. It is also known as messenger RNA. So what is its function? It transfers genetic information, information from genes to the ribosome, from genes to ribosome for protein synthesis. Okay. Second point is it is synthesized from heterogeneous nuclear RNA. It is synthesized from hnRNA. Synthesis source is hnRNA. hnRNA helps in synthesis of mRNA. Okay, let us talk about tRNA. tRNA, or also called as transfer RNA. Okay, its structure is, or we can say, structurally similar to clover leaf model. Structurally similar to clover leaf model, which having 4 arms. Acceptor, anticodon, D, okay, these are the 4 composition. So its major function is transfer of amino acids to mRNA for protein synthesis. Okay, last RNA is rRNA, also called as ribosomal RNA. What is its role? It helps in binding of mRNA to ribosome. Binding of mRNA to ribosome. Okay, then this is all about, this is a small video which is all about the DNA and RNA. Let us discuss something about mutation. Okay, so what do you mean by mutation? Mutation means if there is alteration in genetic sequence in DNA that leads to development of mutation, and sometimes mutation leads to development of cancer. Okay, mutation may be due to substitution. Mutation may be due to substitution, that is, one base is replaced by another. One base is replaced by another, and this substitution is of two subtypes. Okay, what are they? One is transition, another one is transversion. One is transition, another one is transversion. Okay, what does transition means? If purine to purine or pyrimidine to pyrimidine substitution is there, purine to purine, that means A may be substituted by G, G may be substituted by A, or pyrimidine to pyrimidine. That is between T and C. Maybe T may be substituted with C, or C may be substituted with T. Okay. Then in case of transversion, purine to pyrimidine, like A, in case of ATGC, in case of transverse mutation, there may be ACGT type of base sequence may be there. Okay, then one is substitution, one may be due to deletion of something. Second one is deletion. So deletion means deletion or insertion of genetic sequence. Deletion or insertion of some genetic sequence. Okay, this type of mutation is a special type mutation called as frame shift mutation. This is called as frame shift mutation. And third one is maybe due to chromosomal rearrangement. Okay, so these are the major cause of mutation, that is maybe due to substitution, maybe due to deletion, or maybe due to chromosomal rearrangement. And one important point here I want to discuss about some start codon and stop codon. Okay, codons are the three nitrogenous bases containing substances which indicate the initiation, start, or stop type of signals. Okay, so there is a only start codon, that is AUG, that is methionine. So methionine amino acid is act as a start codon. Okay, stop codon is basically we have three stop codons that are UAG, UAG, UGA and UAA. UAG called as opal. Okay, UGA is called as amber and UAA is called as ochre. Okay, these start codons we are also called as sense codon. This stop codon we also called it as nonsense codon. Okay, so when the start codon is bind, the transcription process carry out, but when the stop codon binds, the transcription process inhibited. Okay, or DNA synthesis process get inhibited. Okay, thank you.